Hey guys, here we are with chapter 10, which is on gene action from DNA to protein. So we're going to start by just discussing a little bit of what this is. We all know what DNA is. After all, this is chapter 10 in our genetics class. But what this chapter focuses on and what we're going to learn about is how you take the DNA and use it to actually create protein. So in other words, the idea of protein synthesis. And this is important because, like we keep saying, the proteins are what do pretty much all your functions. They run everything, they take care of everything, they are super duper essential. So now we're going to talk about how you go from the DNA, specifically a gene in the DNA, and use that information to create a functional protein. This is section 1, which looks at the first step of protein synthesis, which is transcription. There are 3 steps in protein synthesis, the first being transcription, the second being translation, and then the third being protein folding. We're going to focus more so on the first 2, a little bit discussion on the last one, but not a whole lot. All right, so shall we get going? Yes, we shall. So section 1, transcription copies the information from DNA. So let's go ahead and get started on this. So transcription copies the info from DNA. Now, as you remember, in your DNA, that's your genetic code. And within there are these things called genes. There are segments of the DNA that code for a particular trait. And what transcription does is it takes a gene in your DNA and it makes a copy of it, but not a direct copy of DNA to DNA. It actually takes it and turns it into something called RNA. So we're making a copy of DNA just in a different form. Now, both DNA and RNA are both types of nucleic acid. We have DNA as deoxyribonucleic acid, and we have RNA as ribonucleic acid. So first thing first is transcription, and that's where we take the information from the DNA and use that to actually create the RNA. After that, the RNA is going to go out into the cytoplasm, where it is going to be used to construct an amino acid chain. Now, proteins are large macromolecules. They're polymers, and all polymers are made up of smaller repeating subunits called monomers. The monomer of protein is amino acids. So what happens is the information in the RNA is going to help us organize the amino acids into a chain, a polypeptide chain that is bonded together, and the order of the amino acids, which amino acids go where, how long the chain is, all of that is going to be determined by the information in the RNA. From there, then that's called translation. From there, we take that chain, we fold it up properly into a functional 3D shape, and that's how we get a working protein. Now, in molecular biology, we do consider the central dogma of this topic, central dogma being the core belief, is the idea that the directional flow of information, it begins in the DNA, then we use the DNA to make the RNA, and then we take the RNA and use that to make the proteins. So this idea that the information flows from DNA to RNA, finally to proteins, that is the core belief, the foundation for everything else in molecular biology. That's why we call it the central dogma. Now, the cool thing about that, like we said, is a lot of times in molecular biology, we talk about how enzymes work, or reactions are run, or how different types of molecules interact, or how antibodies work and all that. Well, guess what's doing all that? Yeah, proteins. And so that's why this is a central dogma, because we're looking at how to make proteins from RNA and how do we make RNA from DNA. Now, there's a lot of different ways that, I shouldn't say ways, but there's a lot of things that regulate how genes get expressed. So gene expression means that we are taking a gene and we are using it to create protein, and the protein is going out and doing its job. So there's many different ways that we can actually control this. So which gene gets expressed and when, how often does it get expressed? Also in terms of cell differentiation. That's when you take essentially a stem cell and you program it to be a particular kind of cell, meaning it's going to have to make particular proteins in order for it to do whatever job it needs to do. So for instance, if it's a skin cell, it's going to have to build different kinds of structures such as hair follicles and build hair and create this sweat ducts and all that. And let's not forget make pigment such as melanin, and in certain amounts. If it's a stomach cell, depending on where it's at in the stomach, might have to secrete different enzymes for digestive purposes. So gene regulation is what tells genes when to be expressed and how often to be expressed. And because of that, it also does play a role in cell differentiation, which is telling a cell what job it's going to be, what kind of cell is it going to be, and thus what kind of proteins it will need to make in order to do that job. So before we move on and talk about transcription, let's talk a little bit more about RNA. As we said, RNA stands for ribonucleic acid. It is a type of nucleic acid similar to DNA, but it is not the same as DNA. There's some key difference, which we'll talk about in just a moment. So what happens is, RNA is actually copied from one of the DNA strands. So remember, DNA is a double helix. So it's two strands that are twisted together. Since these two strands are twisted together, what happens is we untwist it, and then we can use one of the strands, which we call the template strand, in order to make the RNA complementary to it. It's a process very similar to DNA replication. That's what transcription is. We'll talk about that again also in a little bit once we're done talking about RNAs. So there are some key differences between RNA and DNA, and not just the fact that the acronyms are different. First off, as we said, DNA is a double helix. So there are two strands that are twisted together. RNA, there's actually a lot of different types of RNA. We're only going to really talk about a few in this section, but there's a lot of different types of RNA, and they have different functions. But most of them are actually single-stranded. So DNA is double-stranded, RNA is single-stranded. RNA, since it's made from sections of the DNA, also tends to be much smaller than the DNA. There's also a little bit of a change in the bases. In DNA, the nitrogenous bases include adenine, guanine, cytosine, and thymine. Well, in RNA, there is no thymine. Instead, you have what's called urocil. So it's adenine, guanine, cytosine, and urocil. Now, urocil is a pyrimidine just like thymine, and it does everything that thymine does. It's the exact same functionality in terms of it bonds with adenine. It is complementary to adenine. It's just that in RNA, we don't see thymine. We always see uracil in place of thymine. And then the sugar in RNA is different than it is in DNA. Remember that, trying to get some things taken care of. So remember that in DNA, which is deoxyribonucleic acid, and that means it's a nucleic acid that contains deoxyribose as its 5-carbon sugar. Well, RNA has a different sugar in it. RNA stands for ribonucleic acid, so its sugar is ribose. So these are different sugars. They both are in the same location. They're both in the sugar phosphate backbone of the assorted nucleotide. They both bond with the phosphates in their own nucleotide to the phosphate in the next nucleotide, and they hold the bases out. But again, just slightly different structural difference. Now let's talk about three main kinds of RNA used in protein synthesis. First one is called messenger RNA, or mRNA. And what this is what will actually carry the information from the gene in the DNA in order to be able to create the amino acid chain. So it stores the information for the particular amino acid sequence in order to build the protein. This is what will be made from the process of transcription in terms of protein synthesis. Now, if you look at the picture here, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11. So they're just showing you 11 bases here. Let's see. So the rounded yellow one would be urocil. They tell us that, which means if we know that urocil takes the place of thymine, and thymine is complementary to adenine, therefore adenine and urocil are complementary. It's rounded there. The green one is rounded. That would be adenine. So this way, if it's on another strand, maybe of the DNA, the adenine would be able to lock with it. Same with this. The blue one would be cytosine, and the red one would be guanine. Now, something else that you need to know about mRNA is we tend to break it up into groups of 3 sequential bases. We call this a codon. So it's 3 bases in a row. These are very, very important because it's the codons, these sequence of 3 bases, that will dictate the actual order of the amino acids. We'll see how they work in a little bit. That's more so in section 2 when we talk about protein synthesis, translation of protein synthesis. But just so you know, and you are aware, we do have, let's say, these first 3 right here, the guanine, cytosine, and adenine right there. All together, they would be a codon. And then the next 3 would be a codon, and the next 3 would be a codon, and so forth and so forth. Next, we have ribosomal RNA, or rRNA. And this is actually a structural component of ribosomes, as you might remember from our cells chapter. Ribosomes are a small organelle in cells that help to create proteins. Well, now we're going to talk about how that is. Again, that's more so with translation, which is section 2. But ribosomes will, when it says it physically supports other structures involved in protein synthesis, what it means is that it's actually going to attach to the mRNA in order to hold it in place, and it's going to provide a spot for the next RNA, the transfer RNA, to line up in order to bring the amino acids down. So it provides a place where the different other RNAs can get together in order to allow the amino acids to bond in the proper order. So ribosomal RNA joins with other proteins in order to become the ribosome. The ribosome is a small organelle that will allow the different different RNAs like mRNA and tRNA to come together in order to allow for translation to occur. And then some rRNAs, or ribosomal RNAs, will also help catalyze the formation of peptide bonds. What that means is, as the amino acids line up, it's going to help them to actually bond, to form a bond between them. The entire amino acid chain, we call it a polypeptide chain. And the bonds that hold the amino acids together, those would be considered peptide bonds. And the rRNA is going to help get those formed. And the last kind of RNA that's really important for protein synthesis is called transfer RNA, or tRNA. It's kind of this weird cloverleaf shape. And what they mean by cloverleaf, if you follow the mouse on the screen, notice it starts here, loops out like one leaf of a clover, loops out like another leaf of a clover, loops again like another leaf of a clover. Now this is kind of exaggerated, so you can see these 3 bases down here. It's not gonna necessarily be as big as that. But what this is, on one end, on the top end, it is connected to an amino acid. And it's going to bring that amino acid down to the ribosome. Now it knows where to attach to the mRNA because of those 3 bases at the bottom. See that right there? Remember, we said that in mRNA, the sequence of 3 bases together makes up a codon. Well, on transfer or tRNA, they have a sequence of 3 bases that's called an anticodon, and it is complementary to the codon on the mRNA. So the complementary codons and anticodon bases from the mRNA and the tRNA will line up, and that will allow the amino acid to be put in the correct placement. Again, talk more about that in section 2. All right. So now let's talk a little bit about these things called transcription factors. Now, first off, we'll talk a little bit about bacterial genes. The nice thing about bacteria, we can mention them briefly because they're so different than ours. Now, not in terms of structure, but in terms of length. Bacteria are prokaryotic organisms. They do not have a nucleus, and their DNA is a lot smaller than ours, which is why it doesn't necessarily need to be in a nucleus, and it's actually circular. So small just kind of loops together to form a circular structure. Now, because of this, they actually have their genes organized into operons. And what an operon is, it's a unit made up of linked genes, which is thought to regulate other genes responsible for protein synthesis. So it's a unit that helps to regulate protein synthesis. And because it has such a strong and consistent role, it is regulated in a coordinate kind of way, meaning that, you know, it's kind of going to do certain things at certain times as needed. We don't see that in our DNA in eukaryotic organisms typically. It's very, very rare for operon organization to occur in eukaryotic organisms. However, it has been found in a certain species of roundworm. Bear with me as I try and say this. Canor habitidus elegans. I probably said that wrong. I'm sorry. So with the exception of C. elegans, it is typically eukaryotic genomes don't have operon organization. Instead, we have these complex set of transcription factors. And there's multiple factors involved, different kinds of proteins and other molecules. And what happens is, depending on the actual combination of transcription factors, that will be what is going to determine which gene is created, or sorry, not created, which gene is expressed and which protein is created. Because of the importance of these transcription factors in gene expression and regulation, it is thought that there are some genes that, some diseases and disorders that occur in humans that aren't necessarily because of defects in the DNA itself, but maybe because of defects in the transcriptional factors. If there's defects in the transcriptional factors, then that means that they may not transcribe and thus translate the genes correctly, which means it could then affect how the gene is being expressed. All right. Let's move on to the steps of transcription. Again, we said transcription is taking the DNA and using it as a template to make the RNA. Specifically, in this case, we're going to talk about how to make messenger RNA. It is similar to DNA replication. Of course, there are some key differences. We're not going to transcribe the entire DNA. We're only going to transcribe whichever gene we're interested in at the time. So we have these different transcription factors, and the combination of the transcription factors that are involved are going to dictate which gene gets transcribed. And then we have RNA polymerase. Now, RNA polymerase, like DNA polymerase is going to come and help us to actually build the strand. We'll get to that in a moment. And what happens is these can recognize a certain region of DNA near a gene, usually near the beginning of the gene. We call this a promoter sequence. And this promoter sequence is very important because it pretty much says, hey, attach here. There's a gene right here, so attach here. So the transcription factors can actually recognize where the promoter is, and they will bind to it. When the transcription factors bind to the promoter, it then attracts the RNA polymerase down to that same spot where it can also bind, and it can actually begin transcription. Now, something I neglected to say, because it's kind of what I meant by similar to DNA replication, but remember, the DNA is all coiled up, so we do need to loosen it. And DNA helicase, we talked about that enzyme back when we were talking about DNA replication. It's going to attach and kind of unwind and unzip the DNA. This is what will allow the transcription factors to actually have access to the promoter region. So once helicase opens up the promoter region, or the DNA double helix, the promoter sequence is now exposed. The transcription factors can bind to it. This will attract the RNA polymerase to now bind to it. And polymerase is going to start reading the template strand, so the template DNA, and building a complementary RNA strand to go along with it. So it's going to add RNA bases that are opposite. So for instance, if it sees guanine in the template strand, it's going to add a cytosine RNA nucleotide. If it sees cytosine in the DNA, it's going to add guanine in the RNA. If it sees thymine in the DNA, it's going to add adenine in the RNA. And if it sees adenine in the RNA, it's going to add urocil. Because remember, there's no thymine in RNA, so it's going to add the urocil instead. So it's going to make complementary RNA bases and attach them together to form the mRNA sequence, which you can now see here. We have the picture, the red and blue up top. Those are the DNA double helix. It is open courtesy of DNA helicase. Let's see. Somewhere around here probably would have been the template strand, the promoter. The red is the template strand. Somewhere around here would have been the promoter. The transcription factors would have bound to it, and then that would allow the RNA polymerase to bind to it, and then it starts copying what's exposed. Just like in DNA replication, the RNA is made in, or RNA polymerase moves and makes the new RNA strand in a 5 to 3 direction. So you see, this is already made. This is the part that's being made right up here. All right. Now, we're not done. Again, eukaryotic organisms are more complex than prokaryotes. So in bacteria, once the RNA is transcribed, it's immediately going to be translated, which is step 2 of protein synthesis. But we don't see that in eukaryotic organisms. What happens is we have to process the RNA. So we're going to alter it or modify it in some way before it leaves the nucleus and goes out into the cytoplasm to be translated. And that's what RNA processing is. It's getting the RNA ready to be translated. It does need to be altered first. So let's talk a little bit about that. So first thing we do is we have our messenger RNA. It is newly transcribed and it is ready to get going. But before it can, we're going to add some things to the front and back of it. So on the front end, near the 5' end, we're going to add a modified nucleotide cap. What this means is we're going to put some modified nucleotides at the front of it, and essentially this marks it as the front. And then we are going to add a poly A tail to the back of it. What we mean by poly A is multiple adenines are going to be added to the end of it, or 3' end, the back portion of it. As you can see in the picture here, we have 5' cap, modified nucleotide cap on the 5' end. And here we have the poly A tail on the 3' end. Now we're not done yet. We still have to cut out these things called introns. Now introns are non-protein encoding sequences. So again, if you're looking at the picture, you'll notice that the mRNA has these red sections and these lighter sections, like there and there. The 2 lighter sections are introns. Introns are sections of DNA that do not code for a particular protein. So what happens is we have to take these intervening sequences, these non-encoding sequences, and we have to get them out of the mRNA. So we're going to cut those out, and then what we're left with are, again in the picture, these red sequences. We call those exons. Exons are sections or sequences that encode for protein. So they're what's going to be used in order to build the protein chain. So we're going to take these remaining pieces, we're going to cut out the introns and take these remaining red pieces, push them together, make sure they're all bonded, and we are left with our modified mRNA, and it has been all processed and ready to go. Now, to be honest, we're not 100% sure on the functions of introns. We will see one function of them later on in this section. Not this section, sorry, this chapter. In section 3, we're going to talk about the introns again and how they can be used to make different proteins. But overall, we're not 100% sure of their full functionality. What is their full job? We know a little bit about them in terms of making different types of proteins, but we don't know everything about everything. That's the nice thing about science. We're constantly researching and getting new information. So who knows what we'll figure out and learn about introns in the future. We have these things called ribosomes, and they're actually these little tiny small RNAs with catalytic activity. And what we mean by catalytic activity is that they help with the cutting out, the splicing of the introns. Now these ribosomes, they join up with proteins in order to form these larger structures made up of ribosomes and proteins that are called snRNPs. You can see that acronym right there stands for small nuclear ribonucleoproteins. And then these snRNPs then come together to form a larger structure called spliceosomes. Spliceosomes are large, complex molecule that is typically found within the nucleus, and its job is to cut out or splice the introns out of the messenger RNA. So ribosomes come together with proteins to form snRNPs, and then snRNPs come together with other snRNPs to form the spliceosome. And the spliceosome is what will actually cut out. It'll edit the messenger RNA by cutting out, or what we call splicing, these introns out of the mRNA, which will allow the exons to be pushed together, reform bonds to form the fully edited and processed mRNA. RNA editing changes the size of the protein product in different cell types because we are taking out sequences of bases. If we are taking out sequences of bases, that means we are taking out codons. Because remember, a codon is 3 bases in a row. And 3 bases in a row, the codon code for 1 amino acid. So if we're taking out codons, And that means we're going to have less amino acids in the final product of our protein chain. So depending on what kind of cell you are, you're going to be making different kinds of proteins, or maybe even different versions of the same protein in order to do the job of whatever type of cell you are. So this editing is a very important process because it does help with the different cell types doing their different jobs, because all your cells need to make different kinds of proteins. The human body makes over 22,000 different proteins. And this helps different cells to have similar but unique proteins that do similar but distinct jobs. All right, so we've edited it. We have our modified nucleotide cap. We have our poly A tail. We got rid of the introns and pushed the exons all back together and rebonded them. We now have a fully processed messenger RNA. And now that it's been processed, it has to be exported, so sent out of the nucleus before it can be translated. Translation is step 2 of protein synthesis. And all the machinery, everything you need to build the proteins is out in the cytoplasm, which means that now that transcription and RNA processing is all done, we have to go from the nucleus where transcription occurs and move out into the cytoplasm in order for the next step to begin. So here's just, again, the picture that we've been looking at in terms of RNA processing. So what we have, we began with just this long red-ish sequence. We added the modified nucleotide cap on the 5' end that delineates this is the front of the messenger RNA. And we added the poly A tail to the end, to the 3' end of the messenger RNA. That tells us this is the end. And then the spliceosome comes in. It cuts out the intron here between 30 and 31, and here between 104 and 105. It's going to completely cut those out. And then these 3 exon sequences that are left, they get pushed together and spliced together. So they're going to be connected into a nice continuous mRNA strand. As you can see, it is shorter than the original one, which makes sense. We cut pieces out. And because it's shorter, less bases means less codons, means less amino acids, which means that the protein that is going to be made is going to be smaller than the initial one could have been. But again, we do this because we're trying to get a particular function. All right, so that is it for section 1 notes. I hope this helped you. If you have any questions, please hit me up in the Google Classroom or through email. At the end of the entire chapter, I will have links to videos that have good review information for everything in the chapter. But until that time, I hope this helped. Again, if you have questions, please let me know. I can't help you if you don't tell me. So I hope everyone is doing well. I hope this helps to explain some of the information. And until next time, everybody, take care of yourselves, all right? Stay healthy, stay well, and